For those of you who are unfamiliar with the company, Oryzon Genomics is a publicly listed company in Spain. So we have to make some mentions to this legal notice. Oryzon developed potent and selective epigenetic drugs in the field of oncology and CNS. We are a specialist in one specific target, which is LSD1, lysine specificity methylase 1, an histone modifier, a chromatin remodeler. And we have a program, phase 3 ready in CNS, Vafidemstat, and we have also a program in phase 2 in oncology, iadademstat, which I will mention very briefly today. So we have an experienced management team, experienced board of directors, and well-renowned clinical, regulatory, and industry experts to help us moving forward to registrational steps. The company raised $60 million last year in equity and grants, non-refundable grants, and has right now cash runway till the first half of 2027. So as I said, we are a specialist in CNS and a specialist in LSD1. We are going to focus today, obviously, in the CNS program, but I would like also to make a very short mention to our program, an independent program against the same target, LSD1 in oncology, where we are leveraging the collaboration with the National Cancer Institute. So before I do that, just let me remind you that Oryzon is right now one of the top 50 public companies in Europe, and certainly one of the most liquid small companies in the whole continent. We have traded more than 270 million shares last year, which we have been flirting with around 1 billion euros trading, which is, as you will see in this slide, on average, every quarter, we trade daily 1 million shares. So this is also representing the last financing we did last year, where we raised 30 million euros in straight equity with no warrants or any other instruments attached. And as you can see, the strong aftermarket performance allows us to have our investor bases very, very happy. So let me just distract you for a few seconds with our oncology program, because I think that it would be unfair to present you the company without mentioning this part of it. So we have an asset, iadademstat, which is an LSD1 inhibitor and a small molecule oral, which is, among other indications, is basically addressing AML, acute myeloid leukemia, which is an aggressive hematological cancer, often fatal, and for which there is a high medical need and a very strong market opportunity. We just presented a few weeks ago at ASH in Florida, in Orlando, the last data that we have in first line newly diagnosed leukemia patients, where we were presenting data with 100% of responses, and from then 90% of complete remission, which are basically comparing with the current standard of care, which is 37% of complete remissions. So this is positioning us in the first line AML competition in a very well position, where we, in terms of quality of responses, 90%, in terms of tolerability, and in terms of biology coverage of the space, we are significantly positioned against competition. But with this introduction, I will right now focus on CNS, which is the main aim of this conference today. We are basically developing another drug here, another LSD1 inhibitor, Vafidemstat. LSD1 is playing a very important role in the CNS development in mammals and in humans. And we know that it's basically involved in several pathological situations where inhibiting LSD1, we can produce beneficial outcomes. So Vafidemstat, which is our LSD1 inhibitor, is an oral molecule, is safe, and we know that the mechanism of action has several aspects. The first one, we produce an overexpression of genes involved on neuronal plasticity. The second one, we regulate by inhibiting LSD1 the capability of the frontal cortex neurons to assess the stress cues and to react properly. And we know that this is happening through the glutamatergic pathway. We and others have produced a very significant evidence, preclinical evidence in different animal models showing that inhibition of LSD1 has a very strong neurological effect, principally in reducing aggression, also in improving memory, in improving sociability. So this is opening really a broad space for clinical development. And this has to be considered because LSD1, beyond its enzymatic activity, is an histone demethylase, is also a critical part of different transcriptional complexes happening in the chromatin. It's also worth to mention that LSD1 inhibition has been proven to basically rescue significant phenotypes in animal models which are representing neurodevelopmental disorders driven by specific mutations. So what you see here on this circle are mutations which are known to cause different syndromes, like the Kleefstra syndrome, like the Kabuki syndrome, but also being involved in large multifactorial diseases like autism, schizophrenia, and others. So this is an interesting angle that we are also exploring. So what is Vafidemstat? Vafidemstat is an oral molecule once a day, is basically very brain penetrant. We have the same the ratio, almost an equal ratio between plasma and CSF. It is very safe. We have dosed already more than 400 people, some of them for very long periods of time. We don't see any safety issue. We don't see drug-drug interaction. And particularly of interest in this space where many psychiatric agents have undesirable effects like weight gain, sedation, sexual dysfunction, or extrapyramidal symptoms. We don't see any of those with our molecule in so far any of the clinical trials that we have performed. So when we have explored on the different clinical trials the angles of memory, the angles of plasticity, the angles of immunoprotection, we finally decide to move forward for aggression. And aggression has to be, it's important to note, aggression is a huge medical need in CNS diseases, and it's a very lateral symptomatology that occurs in many neurodegenerative and psychiatric disorders. So we have a number of clinical evidence that Vafidemstat can rescue aggression and agitation on those patients. And that's why we are focusing right now on trying to apply this angle on different clinical trials. Our clinical development so far, as you can see here, is first and at most is in borderline personality disorder. We were recently ending a phase 2 trial with very interesting results. And we are right now starting the preps or working on the preps to start a phase 3. I will talk about that in a few minutes. We are also having an ongoing schizophrenia phase 2 trial, and we are about to start a phase 2 trial in autism precisely focused in aggression. Okay, why aggression? Besides that there is, as I said, a common feature in many CNS disorders, we have a seminal observation through a basket trial, a phase 2a trial that we ran some time ago with 36 patients from ADHD, from borderline personality disorder, and from autistic spectrum disorders. All those patients were having a significant degree of agitation and aggression. When we treat these patients for 2 months, what we saw was a significant decrease of this agitation and aggression status by all the scales that we use in this trial. So we decided to move forward in this direction, And the first angle that we wanted to explore was, or the first indication we wanted to explore was borderline personality disorder. Why? Well, BPD is a very prevalent disease. We are talking here about 9 million people aggregated between the US and Europe. It has two main symptomatologies, a symptomatology more psychiatric, where we see problems from those patients to have interpersonal relationships, but it has a strong component of agitation and aggression, including self-aggression and suicidal behavior. The question is that for those patients, nowadays, there is no drugs approved. And these patients have been treated with off-label antipsychotics. We have, as we showed you before, preliminary data that shows a clinical signal on reduction of aggression in BPD. And we also know that in preclinical models, it works, and the mechanism of action makes an explanation why it should work. So with all this, we prepare and perform a global phase 2 trial, which we performed in the US and Europe. Was a randomized, placebo-controlled, randomized 1-1 with 211 patients, in which we were basically measuring aggression, overall improvement, and other secondary endpoints. When we went to the FDA, we had the problem that everybody, any sponsor has, facing a regulatory body in an indication where there is nothing approved, which is what are the endpoints that regulatory, from the regulatory point of view, make sense to assess the improvement of those patients under the pharmacological treatment, because there is nothing so far proved and nothing so far has worked in this area. So cognizant of that, we were discussing with the agency, and we decided to duplicate the scales to assess the same observation. So we duplicate the scales for aggression, we duplicate the scales for overall improvement, and so on and so forth. I'm not going to show you all the data of the phase 2 trial that we were releasing in this same conference room last year. I'm going to just mention a few graphs. And as you can see here, in terms of aggression, we were using the classical CGI, which is not a real scale, it's a clinician-rated questionnaire, where basically the doctor asks the patients how you have been feeling on the last 3 months. The patient says this or that, and then the doctor rates from 1 to 7, from less to more acute status. And as you can see, although we saw a better performance of the patients treated with the drug, this better performance did not achieve the necessary clinical statistical significance. Well, that in any other indication would have been the end of it. But in this case, on this particular indication, and that's why we were discussing with the FDA the duplication of scales, we were having a special attention to the other secondary endpoint, which is the exact-situate anger, a patient-rated scales, where the patient is basically answering a set of questions, basically 10 questions, and has a much broader dynamic range. In this scale, as you might see here, we saw a clear separation between the two arms. In the week 10 and in the week 12, we were getting clinical significance below P005. And in this period, we were getting 90% of improvement over the placebo trial arm, and 60% of improvement over the placebo arm. So very, very interesting, very safe. And also we saw a significant observation that the episodes of self-injury were heavily reduced on people treated with our drug. Okay, so that was interesting, especially because we did, during the clinical trial, a qualitative research study on the patients and on the clinicians, and the patient rated that the most cumbersome symptoms on their daily lives were basically rated with aggression and anger. So with all this, we went to the FDA, and in a phase 2 meeting, we presented data, we asked them what we should do, we should close the program, we should move ahead, what you think. They were very supportive for us to move ahead and present a phase III protocol with a number of caveats, specifically to include a clinician-rated scale to measure aggression, to do qualitative research, to do a number of psychometrical analysis, and so on and so forth. What we did, and when we present the protocol following these recommendations, we got a bit surprised, because at the end, we saw the feedback we received from them is that we need still to keep working on that. They are not still satisfied, so we are going to do this on the following months. So to help us on that, we have, of course, the help of the best of the US security, starting by Professor Schasberg, who has been for many years the head of the psychiatric department here at Stanford, but many other very prestigious psychiatrists. But we have also titans of the industry, like Raymond Sanchez, who was the former chief medical officer of Cerebral Therapeutics, or people like Chris Breder, who has been medical officer at the FDA for many years, and we recently incorporated as well Iman Barilero, who is our senior regulatory advisory regulatory affairs expert. So with all this, we are refining this phase III that we are going to present to the FDA as soon as we complete all the extra homework that they have requested to us, which is going to be around 350 patients randomized 1-to-1, and which is going to be as primary endpoint measuring aggression by STAXI-2 Tanger, and including a key secondary clinician rated, which is going to be the OSM. Okay, so let me move forward. This is the main program of the company. We are going to be the first company in phase III in borderline personality disorders, huge medical need, huge population. But we are also very interested in expanding our aggression program, and particularly in autism. So we are going to start a pilot program in Phelan McDermid Syndrome, and we expect to expand this in idiopathic patients, all of them, to measure aggression. So let me just a few lines to say that we and others in preclinical models have demonstrated that LSD1 inhibitors are able to rescue many of the phenotypes that you see in human patients. So we have seen this in a number of different animal models using our drug Vafidemstat, using other of our LSD1 inhibitors. And what you see here in a very dense populated slide is basically that we are able to increase the sociability of the mouse model, that we are reducing the grooming behavior, the repetitive behavior in a very statistical model. We have also seen a reduction on the aggression and a restoration of the electrophysiological deficits. That is important because when we go at the genetic level and we see what genes are we modifying, what we see is that the genes that we are increasing are, of course, the genes involved in stress control, which are basically responding to the strategy, to the stress issues, but also genes like MPAS4 or DOC2A, which are basically involved in the one of the well-known cohorts that are causing autism, like the deletion of the 16p11.2. So, I mean, there is preclinical data, there is a genetic molecular data, we have clinical data reducing aggression on this population, so I think that we have a very strong foundation. Just to wrap up, I have 30 seconds only just to mention that we are also running a trial in schizophrenia. We are focusing in negative symptoms. This is a trial which is going on in Europe right now. It's a randomized trial, approximately 100 patients, a randomized 1-1, where we are basically measuring improvements on the negative subscale of the PANSS. And we are also exploring the positive symptoms, and we are also exploring cognitive impairment. So this is a space where there is lots of activity. We have seen this morning talking about the muscarinics, about all these new mechanisms of action and new players. We think that we are, with LSD1 inhibitors, contributing to enlarge this very much need for new targets in CNS disorders. And with that, I would be very happy if we still have 1 minute for any questions you may have. Thank you.